Hello everyone, welcome dear students, PhD candidates, postdocs, PIs, professors, friends and family of the faculty. It's my great pleasure to open the fourth edition of BioRevolutions. My name is Ruperto Carola. I'm the Dean of the Faculty of Biosciences. The BioRevolution series introduces new colleagues in the faculty, and this is always a highlight in the life of a faculty. And today our pleasure is to welcome our colleagues Yasin Dagdas from the Center for Organismal Studies and Wiegert, the head of the Neurophysiology Department at the Medical Faculty of Mannheim, who associated with us very recently. Now, new faces always mean new research directions, new teaching approaches, and that make the life of a faculty really dynamic and innovative. And innovation, shaping the future, is a task of a university. This is what a university should do. It's the place where knowledge is transferred from the lecturers to the students, or you may say from old to young, and where, most importantly, this knowledge is being generated. We are the ones who should shape and intellectually prepare and invent what is going on in the future. And one foundation of this innovation is the freedom of science. Freiheit der Wissenschaft in German. This is, in my view, the promoter for innovation. And I was reminded of this last week during the sort of examination of our university by an international panel of experts for the Excellence Initiative. One reviewer asked how we prevent political influence into our work and research. And the answer was, the freedom of research and science is a constitutional right in Germany. So we are in a very, very good position. We are privileged. And I would like to take this opportunity to remind us that in a democracy, every privilege comes with an obligation. And this obligation is to give something back. Freedom of science means we can basically investigate what we want. Nobody tells us how to teach, and we even get funded for this. Although I must say we could get a little bit more funded than we do. But we have to give something back for this privilege. We are asked to improve our research, to really investigate, to invest into teaching, and I would say we are the right place for this at our great university here in Heidelberg. And with this spirit, I hand over to Professor Alexis Mezel, the Pro-Dean for Research of the Faculty of Biosciences, for handling and mediating this event. Thank you very much.
Thanks a lot, Rüdiger. Thanks a lot. So also welcome from my side. I'm very, very happy to see so many of you, especially younger faces. This is great, super nice. I would like to give you a little bit of a background about this event. It's the fourth edition of this BioRevolution, and the subtitle is New Faces. Those are the new appointed professors that present their visionary research. That's the new ideas of the title. And also put the emphasis on the importance of basic research in addressing some of the societal challenge that we need. And that's the new horizons of the title. So today we are joined by Yasin Dagdas and Wiegert, and both of them will give a presentation with about 20 minutes. And you will have the opportunity to ask them questions, but unlike a traditional talk, we don't ask questions immediately after each of the talk. We hold on to your questions to the last part, so after the second talk, and then we'll have an interactive session at this moment. Okay? So I would like to give a few words about our first speaker, Yasin Dagdas. So Yasin investigates the evolutionary origins and molecular logic of selective autophagy in plants. So what is autophagy? That's a process that safeguards cellular homeostasis, shape developmental decisions, and enhance stress resilience. It's a process, you will hear about it, how cells can digest themselves purposely, and how this helps the plants and the cells to better address changes in the environment. So what he does in his team is he uses state-of-the-art molecular genetics, imaging, and proteomics to reveal how plants maintain and protein and organelles quality under fluctuating conditions. So Yasin did his undergrad studies in molecular biology and biotechnology in Turkey. He moved then to the UK in Exeter for his PhD that he obtained in 2013 before starting a postdoc at the Sainsbury Institute in Norwich, still in the UK. And then in 2017, he moved to Austria, to Vienna, and he joined the Gregor Mendel Institute, where he was a group leader, before we managed to lure him here to Heidelberg, to our greatest pleasure, and join us in 2025 as a professor here at COS. And we are very, very happy to have him with us. And without further ado, I would like to leave the stage to Yasin and tell us all the exciting things about autophagy and plants.
Thank you. Thank you, Alexei. Okay, so thanks so much for coming. And what I'll try to do today is kind of to tell you a little bit about how we try to do our research, what is the motivation, and also kind of very briefly some of our discoveries. But I think what is really important and what I hope you will get from the talk is kind of our motivation to study cellular quality control. And this is a picture that I kind of took on the Altstadt in Heidelberg. You can see this kind of leaf slowly dying. And what this reminds me is kind of the ubiquity of turnover in life. You are constantly degrading things, you are constantly making things. Just to give you some numbers, just like more or less all of us, as long as I hope we are all healthy, we would have kind of built 250 billion red blood cells in our bodies. And we kind of more or less degrade every single mRNA molecules that we synthesize in our bodies. So this turnover is a major part of the life, and this is happening because we get kind of pathogens or just the general wear and tear, but also unlike engineering, biology is not perfect, life is not perfect. A lot of the things that happen in our cells, in our bodies, are simply kind of prone to having errors, and this also requires the turnover. And generally, kind of you could define aging or let's say cellular health by simply this turnover rate, how quickly you can identify these issues in your cells, in your bodies. But of course, degrading all these things is costly. It is not easy to kind of degrade a whole organ because there are simply issues, metabolic costs, and also you cannot degrade, for example, your brain, or you cannot degrade a lot of things in our bodies without any consequences. And these kind of the balance or imbalance of this leads to the aging process. And what we try to do, and this kind of what hopefully this illustrates, is we are trying to understand how these processes are working and help kind of tilt the balance towards promoting cellular health. But this is kind of a very simple diagram, but the cell is really a messy environment. It's a really, really dense, complicated environment. I hope you could take a moment to just picture these numbers. These are real numbers, and this is from a book I highly recommend, especially the young people in the audience, to have a look at this. Within a very, very small volume, 1 micrometer cube volume, you would have 50,000 ribosomes. And all of these contents, all of these ribosomes, proteins, organelles, etc., needs to be constantly remodeled to be able to live and respond to the changes in the environment. So what you have is basically a cellular environment looks like this. It looks very crowded, very messy, and the challenge is how do you identify those damaged or unwanted macromolecules selectively without causing kind of a general cellular disruption. And this is not a simple task, and this is not a simple problem, and we are trying to understand kind of how this happens at the cellular level. And this is kind of happening on a regular cell, but we know that plants are also incredibly good at adapting and living in very, very different environments. I mean, biology happens in the realms of physics and chemistry. all the physical rules, all the chemical rules are governing biology. And you could imagine life in, in these Arctic environments or in these desert conditions is very, very different. You are facing very, very different challenges. I hope that next time when you kind of see a plant, and we see it all the time, when you complain about how difficult your life is, imagine life of this guy, right? There is a car going through, you basically don't look at it and just press on it, but despite all these challenges, these plants are surviving and doing pretty well, despite all the things that we do really hard to kind of make their life difficult. And what these quality control pathways or these turnover pathways are doing is basically modulating the cellular environment so that you can respond efficiently or you can kind of stay in tune with the conditions that is required. And when these processes are not working, in our cells, we get all kinds of diseases, so neurodegenerative diseases, metabolic diseases, etc. When quality control pathways don't work, you get aging and all these diseases. And similarly, plants also get these kind of, they don't do well. What we do here in this kind of experiment is we grow a normal plant, a wild-type plant, and a quality control mutant plant, and we kind of grow them in the Swedish spring conditions. The reason why we pick those conditions is the temperature goes from 0 degrees in the morning around 8 o'clock till 30 degrees by 12 o'clock. And then this happens kind of on a daily basis for several weeks. And we thought, this should require this cellular remodeling, and if quality control is really, really crucial, these plants should not do very well. And you can see these plants are basically dying. They are not really kind of accommodating all these changes that are happening in their environment. And we want to understand how you can do this kind of in a specific and selective manner within this really, really crowded cellular environment. So the kind of barbecue of our lab is to understand how this quality control process called autophagy mediates the cellular quality control process and reprogramming. And the way we are trying to kind of address this question is, this is a universal problem. Every eukaryotic cell will have, proteins kind of becoming aggregating or organelles aging over time. Are they using the same set of tools, or did they invent different ways of doing this? And so we try to kind of comparatively study these processes to see how kind of conserved and diversified these processes are. But in essence, what we really want to understand, and the reason why this is kind of in Comic Sans is because this is the child version of what we are interested in, is we had the kind of last common eukaryotic ancestor, and we have an incredible diversity of all these modern cells. How did all these different cellular innovations arise during evolution? And this is kind of what we want to understand and hope to discover over time. What I will try to show you today is we are kind of how we use evolutionary cell biology approaches to discover new pathways. What you are seeing here is kind of the tree of life. And you can see on this part here, we have different kinds of plants. And we sample these plants to identify different autophagy mechanisms. We also use it for validation. So some of these processes we study, for example, in human cells and also in kind of different plant model systems. And if we are seeing kind of similar results, this indicates that this is a highly conserved mechanism. And I will show you at the very end one kind of crazy example, how we can use, how we can learn from one organism and use it to kind of improve stress tolerance in a different organism. But before I get into our findings, so very briefly, what is autophagy? It is a membrane trafficking process. So you have a de novo membrane forming completely from scratch. So you get these different kinds of cargo that is engulfed into a vesicle that is originating from the endoplasmic reticulum, and then you bring it to the vacuole for degradation. So you kind of eat parts of the cell in a selective manner using a membrane trafficking process. And the selectivity is coming from these modular proteins called selective autophagy receptors that we kind of study quite often. So these selective autophagy receptors specialize on different cargo, so whether it is chloroplast, mitochondria, protein aggregates, or for example, an invading virus. And they directly interact with this ubiquitin-like protein called ATG8, and that is how they kind of make the process selective. So they kind of breach the cargo with the growing vesicle, and that is how the process becomes selective. And before I kind of get into the nitty-gritty details of autophagy, it is just to illustrate you, it is important for plant survival. So for example, this is one of the model systems that we study. It's called Marcantia. It's a liverwort species. You can see these plants are kind of, when we remove autophagy process, they are highly sensitive to nutrient starvation. Or another model species called Arabidopsis is highly sensitive to viral infections. And it is also important for cellular development and regeneration. So when we look at autophagy mutants, they are not really doing very well when we ask them to regenerate. And the same is true for another species called Physcomitrium. So you can see normally when we cut these plants from the edges, they regenerate new cells and organs. But when we mutate autophagy process, this is not happening anymore. So the main unknown in our field, in the plant autophagy field, was the selective autophagy receptors. And this was not simply a kind of blast problem. We couldn't just take the human proteins, find them in plants, and study them. So plants did invent new ways of doing selective autophagy. And to find it, we developed an autophagy discovery pipeline. And we did 3 independent screens using this discovery pipeline. And I'll just very briefly show you what these screens are. So in the first one, which we call evophagy, so we basically said, if you look at these autophagy receptors in 5 different plant model systems that covers more than 1 billion years of plant evolution, do we get the same set of proteins, or did they invent different ways of doing it? So, what you have here is, for example, this is Chlamydomonas, this is Marcantia, you have Arabidopsis, tobacco, etc. And in the virus screen, we took advantage of the viruses that they remodel different organelles to be able to colonize our cells. And this is also true for example, coronavirus. They go and kind of colonize on specific organelles, and they completely remodel them. You can see, for example, these are all induced by viruses when they're infecting your cells. We thought this is a perfect way to damage an organelle. If you want to find mitochondria degrading pathways, we will use a virus that remodels mitochondria, or chloroplast, or endoplasmic reticulum, etc. And the last kind of project that we will continue doing it here in Heidelberg is looking at regeneration. So when you regenerate, and plants are incredibly good at regeneration, what you do is you completely change a well-differentiated cell into a stem cell-like cell, and then they differentiate into new organs. And this is very, very similar, at least kind of conceptually, both in animals and plants. We thought, autophagy could be important for this process because you are undergoing an extensive cellular remodeling. Maybe autophagy is required for this. Just to illustrate how this kind of process is activating autophagy, so when we do cut these Arabidopsis roots, you can see within a few minutes, we get this very nice induction of autophagosomes. All these little puncta contain parts of the cell to be able to regenerate. Because if we now, for example, if we cut these roots in a way that they don't regenerate anymore, we don't see induction of autophagy, but we see induction of autophagy only when the roots are actively regenerating. And this is also true for another kind of regeneration system, which is called callus formation. So in this case, you really kind of regenerate somatic cells into a stem cell-like mass called callus, and autophagy is induced in those cells that form the callus tissue. And similarly, like I showed you before, if you remove autophagy process, this is not happening anymore. And what we are kind of trying to understand now is to see how autophagy process is involved in kind of promoting this regeneration process. Just to summarize, just to give you some kind of examples of what we have identified using these kind of comparative screens. So what we have on the left-hand side here is kind of called orthology. So we are simply looking at the sequences of the proteins and seeing how similar and how different they are. On the right-hand side, we have physical association with kind of using our data sets. So when we look at this protein here, which is called YHBC, it is conserved, like sequence-wise, conserved in all the species that we looked at, but we only identified it in one species. So when we look at it in detail, what happened is during evolution, in that species, this protein acquired a few residues, literally like 4 residues, and that turned that protein from a regular kind of ribosome-related protein into an autophagy receptor. So this is kind of how you can evolve a new receptor from an already existing protein. On another case, we have these 2 proteins that we named as von Willebrand proteins. They are conserved in a group of plants called bryophytes. And when we do phylogenetic analysis, we realize that these proteins originate from fungi. They were literally found in fungal genomes, and over time, with a process called horizontal gene transfer, they came to the plant genomes, and they even started working as autophagy receptors. And interestingly, these proteins are very, very similar to von Willebrand proteins in our cells that are mediating or that are important for blood clotting. So we are now trying to understand what these blood clotting proteins are doing in plants. But for me, this is also a very interesting example where you horizontally transferred one gene from fungi to plants, and that kind of gave them a new feature. And finally, we had this kind of protein, these proteins that were so far uncharacterized, but highly conserved, and we started characterizing some of them. One of them, we called it Caesar, so it binds to these ubiquitinated protein aggregates, and as you can see, when we remove these proteins, these plants are not really doing very well when we kind of stress them with increased heat. So altogether, what these kind of examples illustrate is we could really use these comparative approaches to identify new autophagy pathways, and also these are very useful tools to kind of characterize them in detail. For the tinkering aspect, the motivation really comes from this incredibly influential essay that was written by François Jacob in 1977. So what he says, and I think this is really, really important for all of us to think about biology, evolution doesn't create something from scratch. It basically tinkers the existing material to give it new features. And that's kind of how evolution operates, right? So it is not an engineering process, it operates via tinkering. So you kind of have already existing proteins, and then you modulate them for different purposes. So we were studying this protein called EDC4 that serves as a receptor for degrading proteins called P-bodies, membranous organelles. And interestingly, this protein is conserved from kind of plants to humans, but only functions as an autophagy receptor in Markantia, not even kind of a closely related plant species. But interestingly, this protein was recently shown to interact with a protein called alpha-synuclein that is causally linked to Parkinson's disease. We were like, if the autophagy process is really conserved, can we then take the Markantia protein, put it in kind of human neural cells, would it degrade this neurotoxic protein? So we kind of did this engineering process, take the Markantia protein, put it in induced neural cells, and we could indeed induce the degradation of alpha-synuclein in human neural cells, suggesting that, comparative approaches not only give us kind of ways of discovering new things, but we could also tinker kind of autophagy pathways for improved stress tolerance. And we have done this now in kind of disease-relevant model systems, and some people decided to patent this, and they will test it in more kind of clinically relevant conditions. With this, I'll stop here and then finish with this take-home message that I think comparative cell biology is a very fruitful approach to identify new autophagy pathways and then also kind of characterize them further. And before I finish, I would like to thank all the people in the lab. We couldn't have done any of these without their kind of bravery and also courage, and all of our collaborators, both here and also in Vienna. And I also want to thank you for your attention.
Thanks a lot, Yasin. Fascinating overview of evolutionary cell biology and how autophagy can help, or plants can contribute to help human and animals in general, which is great. Thank you. So we'll move on to the second speaker, who is Simon Wiegert. And Simon is the chair of the neurophysiology at the medical faculty in Mannheim. And he's also the co-director of the Mannheim Center for Translational Neurosciences. And he is, as mentioned by Rüdiger earlier, a co-opted member in our faculty. So what is a co-opted member of the Faculty of Biosciences? So it is not a second-class member of the faculty. I would say it's even first class, because although Simon belongs to a different faculty, he was brave enough to come here and actually teach with us. And I think it's particularly relevant for the student in the audience that we have a very liberal policy in our faculty. Doesn't mean we accept anyone. We're very selective, but we welcome anyone motivated who can significantly contribute to the teaching in our faculty. So Simon is one of those examples. And maybe a few words about his research before giving a few words about his scientific vita that heavily relies and connects to Heidelberg. So his research focuses on the mechanisms through which neural circuits encode, store, and adapt information. And his team uses optogenetics, I think he will show, in vivo imaging and electrophysiology to dissect the plasticity and the functional connectivity in neural networks. And he has developed several methods to control and monitor the activity of neurons. And his ultimate goal is to understand learning, memory, and brain disorders. So as I said, Simon has very strong bonds to Heidelberg. I overheard that he took one of his first lectures in this very lecture hall as an undergrad a couple of years ago. So he's a local. He studied here in Heidelberg, and he graduated in 2009 in neurosciences. After, he went to Basel for a postdoc and moved in 2012 to Hamburg as a senior postdoc, then group leader, and finally as a professor in 2018. But then the call of Heidelberg and the south of Germany was stronger, and he joined back Heidelberg University in 2022 to our greatest pleasure, and was quickly after co-opted in our faculty. So I'm very happy that Simon is here today and will tell us about his work. Thank you, Simon.
Yeah, thank you very much, Alexi, for this very kind introduction. And yeah, as you said already, actually, I feel a bit old because I realized that it was 25 years ago when I had my first lecture in this very place here. So yeah, as you said already, I'm affiliated with the medical faculty in Mannheim, and actually this means, living in Heidelberg, that you have to commute to Mannheim and back every day. So it's actually quite pleasant when I have duties here at the faculty, because that makes the commute, of course, much shorter. Anyway, so in order to navigate in our environment, of course, it might be helpful to have a brain. And actually, one brain area that's quite central to this is the hippocampal formation, which we need basically for successful spatial navigation. Somehow I have some problems with the pointer. I'll just go to this one. Okay, so so the hippocampus is basically required for successful spatial navigation and in general goal-directed behavior, and it basically forms episodic memories, which often also have some contextual and associational aspects to them. But not all memories are the same, and sometimes you can form very rapid memories of pleasant or rather aversive events, especially if you take the bike. And these memories often come also with some unexpected events or with some surprise. And for these memories to quickly form, we have basically another system in the brain, the neuromodulatory systems, which are shown here, the cholinergic system, the noradrenergic system, the serotonergic system, and the dopaminergic system. And all these systems together, they mostly reside in the brainstem, and they form the ascending reticular arousal system. So you can imagine that it's important to have balanced arousal in order to perform optimal in our world, to not get overexcited by some maybe unexpected events, or not basically too, let's say, drowsy or relaxed, so that your performance degrades. And of course, this is not only relevant for us humans, but for many animals in general. So if you imagine, for example, this mouse, which has to forage for food, it basically has to be in an optimal arousal state so that it is motivated enough to go for a food source, to also memorize the path to the food source and the location of the food source, and at the same time to be alert enough also to avoid potential predators like this little cat here. So, and also here we have the same arousal systems in the brain, and these areas then influence the cognitive areas of the brain, for example, the hippocampus. So, how are these environmental features now represented in the brain? To put it very simple, we think that there are neuronal populations that represent certain aspects. For example, these green cells here are called place cells, so they actually map the path of the animal to the location of the food source. Then there might be other cells that represent rather the actual reward itself or the location of the food, and so on and so on. And if they are involved in memories, we also call these cellular ensembles engrams. But how they come about and how they are regulated, how stable they are, is still not very well understood. But it's pretty clear that also here the arousal system and the neuromodulators that I talked about are quite important. And also here you need an optimal state of arousal for these engrams or cellular ensembles to efficiently form. So from this, the central questions of our research are: how are neural representations of the world processed in the networks of the brain, and how are these synaptic and neural networks shaped by these neuromodulators? So now here I talked about synaptic networks, and if you look closely, you don't see synapses here, you just see symbols of neurons, and of course the neurons then communicate to each other through synapses, so they're connected in an actually complex synaptic network. And in the human brain, for example, we have about 100 billion neurons, which are connected by 100 trillion synapses, roughly. And you can imagine that this is quite a complex network when you already look at 4 presynaptic neurons connecting to 4 postsynaptic neurons. And if you look at this matrix, you can see that there's already quite a big complexity that you can achieve simply by changing the connectivity patterns here. But on top of that, synapses, they are just not binary, they are plastic, so they can change their strength. So synapses can become stronger by long-term potentiation or weaker by long-term depression. So that means also the strength of the synapse basically influences here the information transfer. And this actually adds to the complexity and determines also how information is processed in these neural ensembles. And also, of course, here, again, the neuromodulators are important because they influence also here the synaptic plasticity processes. So now I talked already about synapses, about neurons, about neural networks, and also about entire brain areas. And this kind of illustrates a fundamental problem that we all have basically in natural sciences, namely that we have to bridge several orders of magnitudes. And this is not only true in the spatial domain, but also in the temporal domain, where we have processes that happen at the millisecond timescale, like action potentials or synaptic transmission, and other processes in the range of minutes, hours, protein synthesis, or even longer, for example, the formation and storage of long-term memories, and so on and so on. So, of course, here are some old school techniques, but ideally we would have like to have a nice technique that allows us to manipulate and read out brain functions across scales with high spatiotemporal precision. And surprisingly, an answer or a suggestion came from somebody that you would not associate with neuroscience necessarily, Francis Crick, and I think I don't have to explain who Francis Crick is. If you don't know, you just have to go outside and look at the DNA double helix model, which he basically described. But you see here in the background a model of the brain. So in his late years, he actually got quite interested also in understanding the brain. And what he postulated is one of the next requirements is to be able to turn the firing of one or more types of neuron on and off in a rapid manner. The ideal signal would be light, okay, then something with infrared light. This seems rather far-fetched, but it is conceivable that molecular biologists could engineer a particular cell type to be sensitive to light in this way. I don't know, sounds crazy, especially if you look at the brain. I mean, if you imagine now, how do we want to get light into this brain here? Because biochemically, the brain is something like a glass of Greek yogurt. I mean, all the lipids and the proteins, they're highly scattering. And that's also why the yogurt is white and the brain is white. So the light doesn't go very far. But in our research, we're not working with the human brain, luckily. We're working with the mouse brain, which is much smaller. So, you can hope that at least the light gets to a certain extent into the brain. And still, the mouse brain is highly conserved. So it contains most of the brain areas that we're interested in, such as, for example, the hippocampus. Okay, but if we get light into the mouse brain, we still don't have light-sensitive cells in the brain. So how do we get the cells now light-sensitive? And now this actually connects a bit to Yasin's talk. If you look very closely, he already showed this little organism here, Chlamydomonas reinhardti, which is a single-celled algae. And this is Peter Hegemann, who basically spent a big portion of his scientific career with this little organism. And basically what is known about this organism is that it can avoid light, so it can do negative phototaxis, which you can see here. So it basically can migrate away from a very bright light source. And this is because there's something called an eye spot, a reddish pigment, which somehow detects the light. And also, it basically was shown that if you knock out one particular gene, this light sensitivity is completely lost. So what Peter Hegemann and Georg Nagel and others in his team then actually realized was that this gene encodes a cation-conducting channel that is light sensitive. And so this is actually great because now you have a light-sensitive cation channel, and if you put this in neurons, this should actually depolarize neurons. So they sent this DNA of this channel then to the lab of Karl Deisroth, and they expressed it in neurons. So here you see a couple of neurons with the channel in the membrane. And indeed, when they shone laser flashes to the neurons, they could spike the neurons with light. So now we have a way to make neurons light sensitive, and it's pretty straightforward. You just basically need to bring the DNA of the single component channel into the neurons, and you can spike them just by depolarizing a little bit. And then the neurons, since they have the machinery to fire action potentials, they can basically spike by themselves. A bit like kids, you just have to tickle them in the right place, and then all the excitement happens by itself. So now, as Francis Crick predicted, we have a tool to actually activate or drive neurons specifically, and this allows us now to test sufficiency. So to ask basically if a certain type of neuron or neuron population is sufficient to drive or to recall a certain information like a memory or a behavior. But very often you also want to know if something is necessary for a particular feature. So in this way, you need to actually be able to turn off the neurons rather than activate the neurons to see if you take out the neuron, does that then basically also remove the feature that you're interested in, like the memory or the behavior. So and this is a bit of a different challenge because now you have to make sure that no excitement breaks through, right? You have to keep the neurons silent and prevent them from spiking. maybe a bit more challenging, as also illustrated here. So, and this you could in principle achieve by, for example, instead of using a cation channel, to use an anion channel, a chloride channel. And this is something we started to explore about 10 years ago with a group of Peter Hegemann, and it turned out that you can mutate a single amino acid, and this dramatically changes the ion selectivity from cationic to anionic conductance. So here you see a recording that I did very early, and it's actually quite nice to see, because if you now electrically depolarize the neuron so that it spikes, by turning on the light, you can now knock out the action potentials and thereby basically inhibit the neuron. If you turn off the light, the neuron starts spiking again. And then in the meantime, we optimized these chloride channels further. We made spectral variants that you can activate with different wavelengths, and also different variants with different kinetics that stay open for longer time, for example, so that you can silence neurons over long time periods. Also, of course, other groups did similar developments, and actually also natural anion conducting channel rhodopsins were then discovered. So aside from developing several of these anion conducting channel rhodopsins, we also worked together with a group in Israel, with Ofer Yitzhar, to generate actually G-protein coupled receptor-based silencing tools, which you can use then to shut off synaptic transmission in a certain area, which allows you basically to inhibit information transfer in one brain region without suppressing the output of the neuron in another brain region. So you don't globally inhibit everything, you just selectively silence the neural connections where you want. And we also made a tool that allows you to both activate neurons and inhibit neurons with different colors of light in the same set of cells, so in the same experiment. Okay, now we have a nice bunch of optogenetic tools to manipulate neurons, but what about readout of neural activity? And also here, Francis Crick had something to say. So he said one way out suggestion is to engineer these neurons so that when one of them fires, it would emit a flash of light of a particular wavelength. The experimenter could then follow the firing of that group of neurons alone. And lo and behold, now we have actually a large array of genetically encoded fluorescent sensors for all sorts of neural functions. So we have sensors for neurotransmitters, sensors for neuromodulators, to which we also contributed a little bit. We have sensors for electrical activity or second messengers. Okay, so now we have a nice toolkit that allows us to manipulate neurons across scales with light, but also to read out neural activity with light. And in the remaining part of the talk, I would like to just give you two examples how we use light to investigate neural function. And I would like to start with an investigation of synaptic function. So just coming back now to synaptic plasticity, I told you before that synapses can change their strength, so they can undergo potentiation or depression. And this has been thoroughly investigated at the single synapse level already, but what is not so clear is actually what happens over longer time periods. So for example, over days. Do the synapses stay in this state, or do they actually even reconnect, and so on and so on. And for this, we actually used slices of hippocampus where we transduce presynaptic neurons now with channel rhodopsin so that we could spike these neurons with light. And you see here like the axonal projections to some postsynaptic neurons. And in these postsynaptic neurons, we expressed now an optical indicator for morphology and an optical indicator for actually synaptic transmission, in this case a calcium indicator. So now when we spike the presynaptic neuron, we can actually visualize synaptic transmission to the postsynaptic cell or synaptic responses, and we can measure the strength of these synaptic responses. But we can also now induce synaptic plasticity. And then, since this is all optical, we can follow these synapses over long time periods and simply ask what happens to these synapses after we induced synaptic plasticity. So, to summarize all of this, what we find is that if we induce long-term depression, so if we functionally weaken the synapses, we see that the synapses have a lower survival rate over time. They're more likely to be eliminated whereas if we induce long-term potentiation, they have a higher stability, so they stay in the circuit for longer. And this is just illustrated here again, where the potentiated synapses basically structurally remain stronger. The depressed synapses are more likely to get lost. And what does that mean? So that means basically if we induce plasticity on the short time scale, on the longer time scales across days, this will lead to changes in synaptic connectivity, because some synapses get lost, some synapses get more stable. And this is now something we're also investigating in vivo in mice during wakefulness, where we use a similar approach to stimulate presynaptic neurons and then record synaptic responses in postsynaptic dendrites and spines. So this is just illustrated here, where we follow basically structural features of the synapses, but also the synaptic responses, and want to now basically confirm in vivo what we observed in these slice experiments. And also to go a step further, or to zoom out a little bit, we can also now look at neuronal ensembles, neuronal representations of space in this case. So here's a mouse that is walking on a treadmill, and it has different spatial features. And while the mouse is doing that, we can image now a big part or a certain part of the hippocampus, many neurons, and we can then see how these neurons map the linear treadmill, which is shown down here, across days. And of course, the next step is then to see how the arousal system of the brain has an influence on the stability and on the formation of these neural representations of space, on these neural maps. Okay, now let me get to the last part, where I just want to show an example at a very different scale, where we look at entire brain regions and not at single synapses. And I just come back again to the arousal that I mentioned earlier. And I said that you need an optimal state of arousal to have optimal cognitive performance. And you can imagine that very well. If you're very drowsy, or if you're maybe too anxious for whatever reason, you might not be able to cognitively process the information that you're now getting. And interestingly, you can read this out actually at the pupil. So if you have a high level of arousal, usually you have a more dilated pupil than if you're very drowsy, then the pupil is more constricted. And if you know your physiology, this basically is connected to the autonomous nervous system, where you have then here a bit too much parasympathetic tone maybe, and here too much sympathetic tone. And this idea is not entirely new. It was actually proposed already a while ago that all of this is controlled by a tiny brain area, the locus ceruleus. So this is the main noradrenergic neuromodulatory system of the brain here in the mouse. Of course, there's two nuclei. This is just one hemisphere that's shown here. And the nucleus, the locus ceruleus actually is a major driver of the pupil, and is actually also associated with exactly what I showed you before. High activity or too low activity is associated with worse cognitive performance. So there's now a lot of direct and indirect evidence in many species that the locus ceruleus is indeed a driver of this pupil-linked arousal that I just mentioned, but it's much less clear how the other neuromodulatory systems that I introduced in the beginning are involved. So, and this is something that we also investigated. So we focused on the two catecholaminergic systems, the serotonergic and the dopaminergic systems, and asked how does the serotonergic system drive the pupil, and how does it interact with the locus ceruleus, and the same for the dopaminergic system. So I will just focus here briefly on this part. And in order to do so, we need a way to selectively target now only the dorsal raphe nucleus, which is this serotonergic system, and the locus ceruleus, which is the noradrenergic system. And so we developed a way to do so, and we can basically then follow the activity of these brain nuclei, or manipulate one and look at the activity of the other, and so on. And we don't do that now here with microscopy, but just with optical fibers that we can implant over these nuclei. The fiber tracts are just depicted here, and this is basically how it then looks. And either manipulate or read out the activity of these brain nuclei. So the first thing you can do is, of course, to just look at correlations, look at the pupil, and monitor the two brain areas simultaneously and see what happens. And what we actually found is that both locus coeruleus and the serotonergic Raphe nuclei are highly correlated with pupil dynamics. So for the locus coeruleus, this was expected, as I said before. For the Raphe, it was not so clear, but there's also a very high correlation to the pupil. So this suggests that there is maybe an involvement also in pupil dynamics. And then you can use different approaches. For example, instead of observing, you can now manipulate the activity of these nuclei with light again. And so we expressed general ROPS either in the locus coeruleus or in the Raphe nuclei, and then drove up the activity of one or of the other and observed again what is the outcome at the pupil. And we could see that both nuclei drive dilation of the pupil. So this, again, expected for locus coeruleus, but confirming that the Raphe nuclei also have a role. Okay, so this effect could then, of course, be direct, or it could be mediated via the locus coeruleus and then go to the pupil. So we also looked at this by now expressing an optical indicator of activity in the locus coeruleus and driving again the Raphe nuclei, and we observed that indeed also the locus coeruleus was driven by the Raphe nuclei, so this suggests that there is a pathway going through the locus coeruleus, but still we wondered if there's an independent pathway, and so with yet another approach, we now optogenetically inhibited the locus coeruleus while driving up the dorsal Raphe nucleus, and this is the last piece of data. So by inhibiting the locus coeruleus alone, you see a constriction of the pupil, which is expected. Now by driving the Raphe nucleus, we see a dilation of the pupil, and now when we inhibit the locus coeruleus and at the same time drive the Raphe nucleus, we can still dilate the pupil. And this suggests actually that there is a pathway that's also independent of the locus coeruleus, so that the dorsal Raphe nucleus basically signals through the locus coeruleus, but also independently to the pupil. And just with this last slide, I want to show that, of course, this also has some relevance because there are many papers and many also reviews that propose that the pupil can be used as a readout of cognitive state and also maybe of cognitive dysfunction. There are many human studies that already give us hints about this association. So if you better understand how the pupil is driven by the different neuromodulatory systems and how the dynamics are, we can get a better idea about the onset, for example, of neurodegenerative diseases or also particular psychiatric conditions. Okay, with this, I would like to thank my collaborators here. I mentioned the people who were involved along the way, or at least I showed them. So part of this was still done in Hamburg. Now this is the crowd in Mannheim. And I want to leave you with this citation. Thank you.
Thanks a lot, Simon. It was absolutely fascinating dive into how brain works from the tiniest scale to the whole system. So I would like to welcome Yasin back on stage and invite you to ask questions. I think that hopefully, like me, you realize the breadth of topics and that are addressed in our faculty. And I think going from subcellular structure and biochemistry, protein or work, and how cells content gets recycled onto signal processing in the brain is really exciting. What I found particularly exciting myself is how for the crosstalk between animals and plant systems through methodologies, but also through concept. And I think this is really, really a nice, beautiful illustration here. So if you have questions, if you have been here before, you're familiar with this gizmo, which I will throw at you if you have a question. So raise your hand, or I see your first one, easy, and you'll pass to the next one, please.
Yeah, so the first question is for Professor Yasin. So there was some discussion about holding a protein and transporting and things like that, or selective quality control. That means is the mutation of certain things which is stopped. That way we can stop the cancer. But this is the very method evolution uses to have more diversity. So by doing selective things, are we not actually hindering the process of evolution, or finding the method to cure cancer is in a way of stopping the evolution?
I mean, we talked before, and you said you are an engineer. So evolution works with chance and necessity. You know, there is no design, there is no goal. You know, you have a bunch of things that then is selected. So I think what we see is the end product, and saying this is the reason and this is why it was selected is engineers' way of looking at biology, which is not necessarily the way we should think about biology in general.
All right, yeah, great stuff. Thank you.
More questions from the audience? Don't be shy. So, meanwhile, I would have a question for you, Yasin. You made this comment that autophagy receptors get basically co-opted from existing proteins, and then through evolution, you may duplicate a gene, and one now is free up to evolve slightly differently. So, what is known? How easy is it to become an autophagy receptor? What does it take?
Not much is known. Surprisingly, not many people thought about it. So for the one that we used to degrade alpha-synuclein, for example, in human neural cells, we engineered using AlphaFold a human version that can at least do one part of the job, but it wasn't enough to become an autophagy receptor. So what it takes to become a receptor, that is not so far understood very well. And I think it needs to be studied case by case. Yeah.
So in that specific example you raised, you were insertion of 4 amino acids or deletions, I can't recall. But we don't know exactly what is 4 amino acid molecularly.
I mean, we know the amino acids, but when we put it, it was just enough to bring one feature, but it wasn't enough to degrade alpha-synuclein.
Okay. Okay. I have 2 questions on the right, so if you could throw Jan and Casper.
Simon, could you briefly explain how spatial information is mapped in the brain, and how synaptic plasticity plays a role in creating these maps?
Mm-hmm. Yeah, so there's basically the concept that in the hippocampus at least, and probably also in other brain areas around it, you have neurons that fire when the animal is in a particular location. So that means when the animal moves in a 2D space, or also on a linear track, you have neurons that only fire when the animal is in a particular location, and they don't fire before or after. So these are the so-called place cells. And then there are also other cells, so-called grid cells, which form really a grid-like representation of the space. And these cells together, they can form a map, so to say, of the environment. And so the problem that with this spatial code is a little bit that it's not very stable. So if you put the animal on the next day in the same environment, it will not be exactly the same cells that fire again in the same location. So there is quite a bit of what is called remapping or variability, and it's not clear at the moment where this is coming from. And so synaptic plasticity could, of course, help now to, let's say, stabilize a certain cell in a certain environment, because if many cells drive the spiking of a certain cell in a certain place, then these synapses that are used at this moment, they can get potentiated and get stronger. So then the next time some synaptic input comes again, it's easier to drive that cell again. So the idea is that if you have synaptic plasticity, that this basically stabilizes the activity of a cell in a certain condition, like in a certain location. Yeah.
And how do you make this consciously accessible? So if I remember my way in a complex parking structure, then I have conscious access to these cells,
Yes.
in a kind of recreate the map?
Yeah. So likely these cells are not where the memory is, where the conscious memory is. These cells are probably really giving you more an idea of the environment. But then you create memories in other brain areas, like in prefrontal cortex, for example, where you then consciously recruit the information about the space and connect it with other experiences that you made. You remember that there were cars. You have more conceptual ideas that you bring together. So so the conscious memory is of course an interaction of many different brain areas where you have different aspects of the environment, also visual information and so on. So yeah.
Hey, Yasin. I was, So I was thinking about the image you showed, that autophagy, that these little puncta of ATG8, I guess, right, really go up when you cut the root. So during normal growth, you also have stem cells dividing and turning over. And for example, when you make a new leaf, you have a lot of growth. So is then autophagy also upregulated in those processes? Are there really clear cell-specific differences in autophagy during normal growth, or is it only really upregulated in stress situations?
There are clear cell type specific differences, and there are also kind of different ATGAs, for example, in Arabidopsis, that responds to different stresses. Just to give you an example, there are cells that form root hairs, and then there are adjacent cells that don't form root hairs. And the cells that form the root hairs would be full of autophagosomes, but the ones that is literally touching them would be kind of black. They wouldn't form anything. So I think it's kind of, it's like the second stage, almost like people have been simply grinding whole organisms, and we are slowly getting into kind of looking into different cell types, etc.
Yeah, so in the case of those root hair cells, then it's really necessary to fuel all that extra growth of the membrane and so on. Yeah. Cool.
Yep. The question at the very back. Excellent.
All right. Another one for Yasin. Maybe a naive question, but how do autophagy receptors actually recognize as large a structure as an organelle? I'm having a hard time visualizing it, that a single receptor sort of...
So it's not a single receptor. So what you do, for example, for mitochondria, when mitochondria gets damaged, you coat it with a polyubiquitin chain. You basically kind of have a coat, and then these receptors have ubiquitin binding domains, and they kind of coat the mitochondria and then bring the autophagosome around it.
In that case, the selectivity is mediated by...
Eat-me signals. You kind of label that organelle as kind of non-self and put some eat-me signals on that organelle, and that is how it becomes selective. Because what you really don't want to do is eat a healthy mitochondria, because you don't have the energy to kind of just waste eating a healthy mitochondria.
Okay, interesting. In that case, there's sort of a functional overlap with the ubiquitin proteasome system. All right, another question?
More questions. Oh, there is one in the front. You have to turn back and touch the box.
Thank you. So I just have a very small question. Are there any disadvantages of studying all these molecular systems at different plant species? Like to study them, are there any disadvantages or limitations?
We are never comfortable. We never feel like experts. But you could also say it never gets boring.
Thank you.
Gaspar.
Thank you. Quick question for Simon. Regarding all these beautiful optogenetic tools in the different colors and inhibitor and activator, are these all based on retina-based generodopsins, or is there any other family?
Yeah, so I mean, what I talked about right now is all retinal-based, and you probably alluded already to a potential problem with the different colors that, yeah, the retinal opsins, of course, you can get a certain spectral range up to maybe 600 something nanometers that you can activate or redshift the sensitivity. The limitation there is, of course, always a bit that you just have the retinal, so it's always also sensitive to blue light, so you cannot truly shift it to red and not have a blue light sensitivity. So this is something that limits a bit the spectral possibilities that you have, also to combine different opsins. I showed this bidirectional opsin bipoles where, I mean, we play a little trick. So we use a blue light inhibitory opsin together with a red light excitatory opsin. And so in this case, you would only have, for example, excitation in the red spectrum and never excitation in the blue spectrum, because there you have an inhibitory opsin. But yeah, they're all, these ones are all retinal based that I talked about.
And do all neurons have retinal? Because I know the fly people need to feed their retinal.
Yes, they actually have retinal. Yes, they synthesize retinal. So it's probably the astrocytes that synthesize the retinal for the neurons. Yes, you don't need to supplement it actually. Yeah. But yes, in vertebrates, often you have to supplement the retinal so that it works properly. Yes.
There is a question in the front, and then we go to the back.
This is for Professor Yasin. I was wondering, are auxins in any way related to autophagy or the process of autophagy?
Auxin?
Yeah.
Yes, we just had a paper on how auxin induces autophagy. Yeah. I mean, auxin is involved in pretty much anything that you can think of, but they use autophagy to kind of induce developmental reprogramming.
Yeah. Thank you.
Thank you. In the back.
Use a relay station.
Thank you. Thank you for the talk, Professor Simon. I was going to ask, you were talking about the optimal state of arousal in cognitive performance. And maybe I'm overcomplicating in my head, but it should be considered the integrations from the other sensory informations. For example, the emotional context, they haven't, because you were talking about the pupil-wide also as the regional reference. But I think this arousal state, how should we define that they are better in this memory performance that you were talking about?
So how the arousal state should be defined?
Yes.
Question? Yeah, I think it also depends a bit on actually what you want to achieve. For example, if you think about this, what I showed, this U-shaped, inverted U-shaped curve for the locus coeruleus. You can, for example, think about different states that you want to achieve. You might want to, just exploit one particular situation. So you want to be very focused on one task and be basically totally dedicated to that task. Or in some other cases, you want to be actually distractible. So you want to be able to switch your behavior, and this is also called exploration-exploitation trade-off. So as an animal, often, I mean, if you find a food source, for example, you really want to exploit this food source. So you don't want to get distracted. You want to be basically focused on that. So in this case, you would want like an intermediate level of arousal, but then, if the food runs out, you want to probably get more into the exploration state, and then you want to become more distractible. And then even if you don't focus anymore on this particular task, the optimal state would be to be distractible and to basically see if there's another food source somewhere around that you can explore. So it depends what you want to achieve, what is the optimal state, of course. Yeah? And of course, arousal is also a bit of a simplified construct in any way, because all these neuromodulatory systems, of course, they have slightly different or quite different functions. and you can't just lump them all together. I mean, now it was a bit simplified. So again, each system also has a bit their unique role. Yeah. So what I just talked about was a bit more the noradrenergic locus coeruleus aspect, for example. But of course, the dopaminergic system is again a bit different. So it might have to do more with really with effort and with dedication to a certain task and so on. And of course, we know that it's also reward related. Yeah. So it really depends a bit what the exact requirement is.
Okay. Yeah. Thank you.
There's a question behind you.
Thank you so much. I'd like to ask both of you a question. In your view, what are the advantages, or are there advantages, specific advantages of conducting biological research in Germany?
Biological research in Germany. Did I get
Germany.
Good place, but
yeah, I mean, I think what we just heard from Rüdiger Hell in the very beginning, I think it's of course a great place, simply in terms of being able and also having funding for doing basic research without any. I mean, you know, let's say in the first place, you don't have to justify why you're doing your research to the funding agency with regard to, you know, addressing a disease or something. You can, if you have a good concept and a good idea, it doesn't have to, you know, lead to a change of society or whatever. So I think this is really good in Germany, that if you have an interesting question, and if it's a well thought through question, you can actually pursue this question. You don't have to treat a disease necessarily. And I think this is a big advantage that we have here for sure.
I mean, maybe I'll give you an example. So one of my friends recently visited a school, and she kind of told about her research. And at the end of her talk, one of the students, so these are kind of primary school students, raised her hand and said, but why are they paying you for? He was genuinely kind of couldn't believe that one could get paid for simply playing. What you have heard today is someone kind of making colorful proteins in mouse brain, shining light, watching them kind of walk in a treadmill, or, you know, we just go grab some organism and study them that in reality has very little relevance to, I don't know, like making our food or something like that. I think that is very, very unique and becoming even more unique around the world. And this is, I think, quite unique in terms of what can be done in Germany. And the second one is the human potential. You know, maybe this will be a kind of, let's say, I'm not trying to, grease you, but we have some amazing people that we get to work with, and bringing them over in Heidelberg and bring them together around the question, that is also quite rare and unique.
Maybe if I may chime in. So the example that I mentioned with research on Chlamydomonas reinhardti, I mean, so Peter Hegemann, he was really just interested in the biophysics of the channel and in rhodopsins. So he wanted to know how rhodopsins work. So he really was, doing a very, very specific and kind of niche-oriented research. But in the end, his discoveries changed an entire field. So in the neuroscience community, he's a hero. And I mean, you know, he did not yet get the Nobel Prize, but he's really close to the level of. I mean, he's very, very well known in the whole field. And I think this is also a good, basically a good example for, you know, what you can achieve or what can happen if you're just allowed to basically tinker and play and be curious. And I mean, this was, of course, all done in Germany by him.
But That's very nice, the point you raised, Simon, because both of you illustrated how through your research you're breaking one of the still subsiding greatest divide in biology, animals versus plants. And you jump from one to the other using techniques or for concept. So what do you think is needed in or the education or into the funding scheme to keep breaking that barrier, which you illustrate beautifully is a great thing to do?
I think you need good colleagues, so that really helps. And hopefully here, for example, in Heidelberg, we will be doing like really kind of operating almost in an organismal barrier-free way. For the regeneration project, we are very keen on working with planaria, working with hydra, working with fish, etc. And kind of also having the right people around that question. And I think we are realizing that these organismal barriers are useful. I mean, you need that expertise, but kind of going across those barriers becomes also very fruitful. You know, you could argue that the biggest discovery that we made in molecular biology is CRISPR-Cas9, and that comes from a bacteria, right? So all these things are, I think, making us more brave to go after a question and see, okay, this organism fits, and we can just use that organism to address that question. But I think you really need good colleagues.
Yeah, maybe an educational system that also, you know, allows a bit of breadth. So, and don't fall for the trap to be more and more and more specialized. I mean, there are programs coming up that are then more and more and more going into just a certain area of research and more focused. And this, of course, you know, takes a bit away your general knowledge or your general way of thinking about things. I mean, I studied biology here in Heidelberg, which was, I mean, I had to, I don't want to say I had to go through botany. I actually enjoyed it, honestly. It was really enjoyable, and I found it super interesting to even do the, you know, the field trips and to draw plants and so on. And no, I think you know, but this gives you a very broad perspective of things, and you're not just becoming an expert right in the beginning. You know, now we have, you know, already bachelors which are highly focused to a certain topic. Of course, then you might be an expert in that area, but you have no idea about general biology. And I think this is actually something that's maybe not to neglect.
Good. This brings me maybe to a more general question that I would have for both of you. So what actually brought you to study biology in the first place?
Well, field hockey.
Field hockey, okay. You dare to explain?
No, actually, I wanted to start biochemistry. So I was already in school. I knew I wanted to do biochemistry. And so I wanted to enroll in already a bit more specialized program. But no, in fact, actually, I used to play field hockey quite competitively. And in Heidelberg, there was one of the centers for field hockey. And so this brought me here to Heidelberg to actually start with biology. So I took the compromise and took biology instead of biochemistry, thinking I can get to biochemistry eventually. But yeah, the path went a bit in a different way, and I don't regret it. So yeah, but I mean, more generally, for me, I was always interested in this research area, and I just wanted to study it. So that was pretty clear.
Okay, thank you. Yasin.
I studied biology Olympiads during secondary school, so I kind of knew I wanted to be a biologist.
The childhood dream came true.
Yeah, and then, you know, I learned about auxin, and I kind of thought that was cool. And yeah, that's kind of continued afterwards.
All right. And is there more questions from the audience? I don't want to monopolize. No, please.
Yeah.
on my brain and saying, okay, now you have to do this.
Well, I guess that's, as you say, it's more a philosophical or an ethical question. I mean, in principle, well, of course, the complex cognitive process of wanting to go here or there, maybe not, but of course, general like cravings for things and so on. I mean, we can already control this in animals, in mice. So you can, you know, there are of course lots of experiments on you know, addiction and so on. And this is, I think, already pretty well controllable in animals and in mice. But for ethical reasons, it will not be so easy to do that in humans, obviously. I mean, one example that actually does work to suppress your appetite for sugars is the SGLT agonists that suppress or that mimic satiety. So this is already a way, a pharmacological way. I mean, of course, I don't think that optogenetics will come to the human very soon. I mean, there are some trials with vision restoration and so on, maybe in the periphery, but otherwise, I think it's going to be very tricky to do that. But it can, of course, help to instruct maybe drug targets or to understand better what type of neurons, what group of neurons do we have to target, and then you develop, for example, you know, a drug that is specific. Yeah, so that would be maybe a way.
So maybe the last question will allow me to take it, and it's directed again to both of you. What are, according to you, the best qualities that a student for biology should have? Any motivational statements for them?
I think biology will kind of shape the future of science in the next coming decades. And we need the smartest and best people out there kind of interested in biology. I would argue probably this is a biased audience anyways, but all the physicists, chemists, mathematicians, informaticians will be doing something related to biology anyways. What kind of people we need? I think we need ambitious people that will kind of put in the effort to go wherever their projects will be taking them.
Yeah, and I think you just have to burn for it. I mean, it's yeah. If you're not intrinsically motivated, and if you study biology to make to make a good living in the end and earn a lot of money, I'm not sure if that should be your prime motivation. I mean, I don't know. You can. It doesn't exclude it, of course. But I think the motivation should really be the biology. I mean, as simple as that. And if that really drives you, and if that's really the thing you want to pursue, I mean, everything else, you know, can fall in place eventually. But I think, I don't know. At least this was always how it went further for me. It was always the intrinsic motivation and the passion for it, and yeah, the rest then, I mean, came along the way.
All right, I think we will conclude this event here. I would like to thank again Yasin and Simon for the wonderful talk. I would like to thank you for your active participation. So there are drinks and some lunches for everybody. If you want to go on and discuss with our speakers or any member of the faculty, please join us. It will be right outside. Thanks again, everyone, and see you on the next occasion.